Hi everyone, so today we're going to be looking at the entirety of chapter 3, which is the 3A and 3B, which is looking specifically at enzymes. So we have been looking at proteins a lot, especially in area study 1. We'll be looking at how they're produced, how we can manipulate them. Most of the proteins that we've actually been looking at and talking about, it's a good chance that the majority of them are actually going to be enzymes. So enzymes are probably the most important protein class that there is. They're involved in the production of other proteins, they're involved in the production of other enzymes, and they play a role in most of the chemical reactions in our body. So we've already looked at some already. We've looked at RNA polymerase, DNA polymerase, we've looked at restriction enzymes, we've looked at them very simplistically. So what we're going to do now is we're going to dive in and we're going to learn more about them because they're going to come up a lot in area study 2 when we're learning about photosynthesis and cellular respiration. So this is the dot point. So this is just tacks on to the first dot point that we looked at, which is just looking at what proteins are. We also need to learn about specifically what enzymes are as well. So what are they? So enzymes are often described as a biological catalyst. So what a catalyst is, it's something that speeds up or triggers a chemical reaction. So how it does is it lowers the activation energy of the reaction, which we're going to look at a little bit later on. The really cool thing about enzymes is that they're not actually consumed by the reaction themselves, so they can be used again and again and again to help facilitate a range of chemical interactions. They are proteins, like we said, and they are specifically cellular globular proteins, which basically means that they can dissolve in water, and globular is just referring to their shape, which we'll look at in a second. This is a really important point here. So the names of most enzymes end in A's. Okay, we've looked at this, DNA polymerase, RNA polymerase. So a lot of them, or most of them, end in the word A's. And this is so handy to know because a lot of the times in exams, they'll probably, they'll give you a question that has no context and they'll be talking about a bunch of different molecules and they'll expect you to be able to understand that it's an enzyme question by giving you a molecule that ends in A's. So it's a really good one to have up your sleeve for specifically exam questions. So this is the general structure of most enzymes. So like we said, they're soluble and globular. So globular just means this sort of weird globby shape here. Normally tertiary or quaternary structures, which is going back to our protein knowledge. So the shape of an enzyme is critical to its function. So hopefully you remember from area study 1 that shape is so important for function of proteins, and that's why we looked at primary, secondary, tertiary, quaternary structure and how those sequences of amino acids and those interactions are so important for the function. And enzymes is a really good example of this because enzymes have a specific active site, which you can see there. They have a specific active site, which is where things bind to. So if that specific shape is not what it's supposed to be, the enzyme's not going to function in the way that it needs to. So the active site of an enzyme is also called a substrate binding site. This is because what goes in there is called a substrate. So a substrate is just a fancy word for any molecule that an enzyme is acting on. It could be a sugar, a fat, a protein. It could be pretty much anything, but it's called a substrate is what goes into the active site. As we said, the structure of the protein is super important because this really specific shape of the active site needs to be complementary to the shape of whatever the substrate is. The attraction between the active site and the substrate is known as affinity, and when the substrate and the active site bind together, they can be called an enzyme-substrate complex. So they're just some keywords you might see floating around, might want to add them to your glossary. So this is what it looks like in a picture, which I think is a lot more useful than words. So our active site is this part here. Okay, it's been simplified in the cartoon, obviously, but this would normally be sort of, involved in that globular structure. So we've got a substrate here. This could be anything. It could be a sugar, fat, whatever. What it's going to do is it's going to bind to that active site. And you'll see here in this second diagram that the active site is completely complementary to the substrate and therefore it is allowed to bind. And this is an example of a catabolic, if I can write, catabolic reaction because it's taking one larger molecule and it's splitting it into two smaller products. So the thing that is released from the enzyme is then called the product. Anabolic would be going the other way. So that's if you have, a smaller substrate and you're putting it into the enzyme to create a bigger product. That's just a good to know, not a need to know, but that's what the enzyme does and what these things look like. So with the enzyme substrate complex, there's two models that are floating around of what that actually looks like. So we'll go through both of them. Both of them exist in the world, so it's good to know about both of them. So we've got this lock and key model for starters. So the lock and key model suggests that the substrate has a shape that is complementary to the shape of the active site. So you'll see here that it fits like a lock and key. So this fits perfectly into the active site and they're both completely complementary of each other. That's what the lock and key looks like, and that's what most diagrams will normally portray as well. But we also have something called the induced fit model, which is probably more realistic because we know things in biology aren't, perfect. So this is where the active site is not an exact complementary shape, but when the bonds that form between the enzyme and the substrate, when they come into contact with each other, this changes the shape of the enzyme so that the substrate can fit properly. So induced fit means that the active site is induced to fit whatever it needs to fit. So you'll see here that the active site isn't exactly perfect, but when the substrate comes over and it the chemistry between them, it changes the shape of this active site so that it is complementary to one another. And you'll see that this is another example of a catabolic reaction, a bigger substrate turning into two smaller products. So it's good to know both different models. So like we said, function and shape go hand in hand, and this is because enzymes are so specific. So each enzyme can only catalyze one particular reaction or a specific set of reactions. So normally one enzyme will be involved in one specific job and it won't really deviate from that. And this specificity relates to its shape. Okay, so that really specific shape from all of those interactions with the amino acids and secondary structures, etc. That's what's causing this really specific shape, and therefore it has a really specific function, and it will normally only correspond to one thing that's happening inside of the body. So this is just a good to know, not a need to know. A lot of exam questions and things like that will give you names of random chemicals and random enzymes, and I just don't want you to be frightened when you see them. They normally won't be relevant to the question anyway, but it's good to be familiar with a few different types of enzymes just in case they pop up. So we've got our intracellular enzymes. So they're the things that they're the enzymes that work inside cells, and some really common ones are things like catalase. We're going to learn about a few in photosynthesis and respiration. So we're going to learn about rubisco and ATPase and all those sorts of things. But catalase is a really big one. So that's what produces hydrogen peroxide, which is harmful in, many metabolic processes produce this thing called hydrogen peroxide, and catalase is what converts this into water and oxygen, which is a not harmful substance. So we're going to learn, we're going to look at that in cellular respiration specifically, but catalase plays a really important function in making sure that those harmful toxins don't hurt us. So that's one that comes up a lot. Then we've got our extracellular enzymes, and they're the ones that work outside of cells. So a really good example of this is amylase, which we're going to be looking at in a prac at some point during term 1 or term 2. So amylase is one that comes up a lot as well. So this is a digestive enzyme. So this is what is in your mouth, in your salivary glands. And this is a really important part of breaking down starches into sugars. So when we eat things, amylase is normally going to be the first one that's going to start breaking down that food. So like I said in the first slide, the main way that enzymes catalyze reactions are by reducing activation energy that's required for them. So catalysts are substances that increase the rate of chemical reactions, and they do this by two main ways. So what they do is they influence the stability of bonds in the reactants. So if you're a chemistry person and you care about all of that sort of stuff, so what they do is they make the stability of the bonds in the reactants either stronger or weaker depending on whether it's a catabolic or anabolic reaction. And they also provide an alternative reaction pathway. So the binding of reactants and enzyme can weaken bonds in the reactants and allow the reaction to proceed more easily. So like I said, it's either weakening bonds or strengthening bonds depending on which way you want to go. And this all reduces the activation energy. So activation energy is basically just the energy that's required to activate any chemical reaction. And like we talked about in Area Study 1, our bodies don't like to use energy, they want to conserve it. So that's why enzymes are so important, because they help us do that. So you'll see in this graph over here that we've got a reaction, and we've got it with or without an enzyme. So this is again a oh, actually, no, sorry, it's not specific at all. We've got our reactants or our substrate over here, and we've got our products over here. So you'll see that without an enzyme, our reaction is going this way. It has a large peak in the amount of energy that is required to complete that chemical reaction, and then it goes down. You'll see with an enzyme that peak is significantly less. So the amount of energy that's stored in the chemicals, or the amount of energy that is required to start that reaction, is less with an enzyme, and that's why they're so important. So you'll see here, and this is what I was getting confused with before, this is the one that splits them into two different types of reaction. So remember I said catabolic. Catabolic is when you're taking a larger molecule and you're splitting it into two smaller molecules. Anabolic is when you're taking two smaller molecules and you're making it into, or joining it together to become a larger molecule. So this is just the same graph, it's just showing that in those two different scenarios. So we've got our substrate or our reactant here. We've got our activation energy here without an enzyme, so you'll see the peak is quite large, and then we've broken it into two separate products, as opposed to having an enzyme, and that peak is a lot lower. And it's the same thing with the anabolic. So we've got our reactants, you'll see they have lower energy to start with because they're two smaller products, and then we've got our activation energy quite large, and then we've got our products here. But with an enzyme, it is significantly less amount of energy that is needed to complete that. So the second part of this, which is 3b, is we want to look at the factors that affect how an enzyme functions. So we've looked at what an enzyme is, how it works. Now we're going to look at the different things that can alter its function, and this is really important in medicine, obviously, and also for photosynthesis and cellular respiration, because a lot of these factors that we're going to learn about now also influence those processes because they're so influenced by enzymes. So the first thing we're going to look at is temperature, and we're going to do a practical look at this as well. But temperature can really affect the activities of enzymes. So traditionally, reactions occur faster at higher temperatures. So all enzymes have an optimal temperature. This is down here. So they all have optimal temperatures, and it'll be different based off of what they need to do. So if it's an enzyme in us, for instance, normally the optimal temperature is going to be about 36, 37, 38 degrees because that's our body temperature. But if it was an enzyme that was working in a different organism, such as like a reptile or something like that, the optimal temperature might be lower because the body temperature of the reptile is lower. So every enzyme has an optimal temperature, and normally an enzyme will work or occur faster, or the reactions will occur faster at a slightly higher temperature, but still within that optimal range. So what happens when we go beyond those optimal ranges? So in this graph here, we've got our optimal range of this particular enzyme. Doesn't say what it is. So you can tell that this enzyme's optimal temperature is about 35 degrees. So you can assume this might be one that's happening in a human or a mammal. So what happens when temperatures get too hot? So when temperatures are too high, denaturation occurs. So what denaturation is basically the breakdown of an enzyme. So high temperatures break the disulfide bonds in the tertiary structure. So hopefully you remember that those are the main bonds that are holding that tertiary structure together. So high temperatures, what it does is it breaks down those bonds. Because it's breaking the tertiary structure apart, this therefore affects the shape of the enzyme, and therefore affects the active site, and therefore the function. Because if the shape is changed, the active site is changed, it can no longer bind to the substrate and it might not be able to function. So you can see here that the enzyme activity severely drops off after this denaturation has occurred, and it's basically useless by the 45 degree mark. From the other end of the extreme, you can see that if it's too cold or colder than the optimal temperature, it's not as severe as denaturation, but you can see that there's an increasingly the enzyme is becoming less and less active as the temperatures lower. So denaturation does not occur for lower temperatures. This is only higher temperatures. But what can happen in lower temperatures is that the enzyme can freeze, which basically means that it lowers the activity of the enzyme. So the enzymes move slower and slower and slower, and then eventually at 10 degrees they stop working altogether. So for temperature and enzyme activity, all enzymes have an optimal temperature. If it gets too high, they might denature. If it gets too low, they might freeze, which in turn will obviously impact the way the enzyme functions. We then have pH, which is a really similar concept. So like temperature, enzymes all have a optimal pH that they work at, okay? So depending on what it does. So you'll see that pepsin, which is an enzyme that works in the stomach to break down foods. So the stomach is super acidic. So you'll see that the optimal pH of pepsin is between 1 and 2 because pepsin needs to work in really acidic environments. You'll then see something like urease, which works, in the kidneys, bladder, etc. So that pH is here, and the optimal pH for trypsin, a different enzyme, is up here. So depending on what the enzyme needs to be used for, will determine the optimal pH for that enzyme. So unlike temperature, enzymes, if they go either way in either direction, will denature. So if the pH is too acidic or if the pH is too alkaline, like in this one here, you'll see that the enzyme denatures in both scenarios. So this pH, this extremes in pH, again, break down the bonds, disrupt the shape and the active site, and therefore it cannot function. We then have enzyme concentration. So this just basically means how much enzyme is present. Okay, so this graph probably isn't any surprise to you. So assuming that nothing else is limiting in this scenario, so assuming that we have, enough substrate there and there's no temperature or pH issues, generally what's going to happen is that if you increase the amount of enzyme, you're going to increase the rate of reaction, because obviously if there's more enzymes present, they are, going to be able to bind to substrate more. They're going to be able to increase the chemical reactions more. So you're going to see a straight upwards direction for that. If you increase enzymes, you are going to increase the reaction rate. And this is always assuming that nothing else is limiting, because if we didn't have, if there wasn't enough substrate to bind to, this is obviously not going to keep going up. It's going to plateau at some point if there's not enough substrate to actually perform these chemical reactions in the first place. Just like I said, substrate concentration also affects this. So I'm assuming that the amount of enzyme is constant and non-limiting, so we're looking at the opposite now. An increase in substrate is going to generally increase the amount of reactions that can take place. So we've got our substrate concentration here, and you'll see that to a certain point, the rate of the reaction is going to increase, because if we have more substrate, obviously the enzymes can bind to more substrate and increase chemical reactions. But what's happening here? Why is it plateauing off in this direction? So a maximum rate is obtained at a certain substrate concentration when all enzymes are occupied by substrate. So eventually there's going to come a time when all of the enzymes are full. So all of the enzymes are bound to a substrate, and there's going to be substrate sitting around doing nothing. Okay, so eventually it reaches a plateau because enzymes are doing all they can, substrate is doing all they can, and this is called enzyme saturation. So when enzymes are completely occupied, this is seen as enzyme saturation, and means that the reactions won't drop, but they will plateau because there's no more enzymes to complete any more reactions. So it's just going to remain consistent. We also have something called inhibition, and there's two different types of inhibition that can affect how an enzyme functions. So inhibition is normally prevents an enzyme from completing its task, which depending on how you look at it can be a good thing or it can be a bad thing. If it's an enzyme that's doing a reaction we don't want, inhibition might be good. But if it's obviously an enzyme that we really need and is really important, inhibition can be bad as well. Depends on the context. So we've got two different types. We've got competitive inhibition and we've got non-competitive, which we'll go through in the next slide. So enzymes can be hindered by molecules known as competitive inhibitors. So they can impede enzymes by blocking their active site. Obviously, if the active site is blocked in an enzyme, it cannot perform its function, so that reduces reaction rate. So it means the substrate cannot bind, no reactions will occur. So the competitive inhibitor is a similar shape to the substrate and allows it to bind to the active site. So let's look at this diagram here. We've got our enzyme. This is our substrate. So this is the normal, natural, what we want to bind to. And this is our inhibitor. So this is what is going to inhibit this binding. So you'll notice that it's a really similar shape. between the two, and obviously the active site is also a similar shape. So what happens is a competitive inhibitor can bind to the active site because it is, a similar shape to the active site. It can bind there, and this binding therefore means that our substrate cannot bind. So if the substrate cannot bind, the reaction does not occur. So it can be inhibited this way by a competitive inhibitor directly competing with the substrate. That's why it's called competitive. So it's competing with the substrate for that active site, and it directly blocks the active site from the substrate, and therefore the chemical reactions cannot occur. We also have something called non-competitive inhibition. So non-competitive inhibitors are not competing with the substrate directly, but they interfere with the enzyme in other ways. So the non-competitive inhibitor, sometimes called an allosteric inhibitor, you might see that somewhere as well. They bind to a site that's not the active site. So they bind to a site other than the active site, which can also be called an allosteric site, which induces a conformational change in the enzyme. So that just means the enzyme changes its shape. We know shape is super important, so if the shape is altered in any way, that means the substrate also can no longer bind. So we've got our enzyme here, we've got our substrate, and we've got a non-competitive inhibitor. So you'll see that it's not the shape of the substrate, it's not competing directly for the active site, but it is going to bind to an allosteric site or a different site in the enzyme. So you see that here. So our non-competitive inhibitor, or our allosteric inhibitor, has bound to an allosteric site, which is just any other site on the enzyme that it can bind to. And you'll see that this binding has changed the active site shape. It has caused a conformational change in that enzyme. Due to that, the substrate cannot bind and the enzyme is inhibited. So the reaction, again, cannot occur. And this is what non-competitive inhibition can look like. Again, can be a good thing if you're trying to control a disease or something like that the enzyme plays a role in, but obviously can also be a very bad thing if that enzyme is involved in a really key chemical reaction. So the last thing we're going to look at is cofactors and coenzymes. So the word co hopefully gives it away. So they are helpers in a way. So they are things to facilitate and help enzymes to perform their function. So they're like assistants, kind of. So cofactors and coenzymes are one and the same, okay? So they can kind of mean the same thing, but a bit different. So we call it a cofactor if it's inorganic. So cofactors are things like ions and inorganic compounds, metals, things like that. We call it a coenzyme if it's an organic compound, like a sugar or something like that. Okay, so cofactors and coenzymes are the same thing, they just have different names depending on whether they're inorganic or they're organic. So cofactors and coenzymes are substances that are essential to the catalytic activity of some enzymes. So some enzymes can function without this, but a lot of enzymes need coenzymes and cofactors to work. So cofactors and coenzymes may alter the shape of enzymes slightly to make the active sites functional or complete the reactive site. So this fits really nicely into that induced fit model. So enzymes that utilize the induced fit model may very well need cofactors and coenzymes to ensure that their active site is the correct shape so that it can bind to a substrate. So coenzymes and cofactors aren't like enzymes themselves. So enzymes can be used and reused again and again and again. So cofactors and coenzymes are often acquired by the body. So a lot of them are vitamins. So that's why it's really important to get them through your diet. And often what they do is they are like ion or chemical what's the word I'm looking for? So we're going to learn more about it in photosynthesis and cell respiration, but basically they're chemical cyclers. So what will happen is you might have something like ATP, which is a coenzyme that we're going to learn about a lot in photosynthesis and cell respiration. So what ATP does, it starts off as ADP, okay, so it starts off in a different form, and then it picks up a phosphate, okay, and then that turns it into a ATP, and then it goes off and it performs its role by then releasing that phosphate to charge a chemical reaction or to help an enzyme perform its function. In cell respiration, for example, would be ATPase. So what would happen is that ADP, it picks up that phosphate, it brings it over to ATPase, which is the enzyme, and it releases those phosphates to help the functioning of ATPase to perform its role. So they're like cycling enzymes. So they're a little bit different to how enzymes work. They're more the facilitators, the helpers. They'll pick up certain chemicals, bring them over. They'll help the enzyme to achieve whatever job it's trying to achieve. So in summary, enzymes are proteins and they're really specific in their shape. And they're really specific in their shape because they need to perform specific reactions in the body. So they catalyze or speed up chemical reactions by lowering the activation energy that is required for it, which is really, really important. They bind to substrates via active sites, and there's two models for how this can occur. So we've got the lock and key, and then we've got the induced fit model. There's a range of things that can affect how an enzyme functions. So temperature and pH need to be optimal, otherwise the enzyme will not perform optimally. Enzyme and substrate concentration can also affect enzyme activity. The more enzyme or the more substrate you have, generally the more reactions are going to happen until you reach a plateau or a limiting factor occurs. Enzymes can also be inhibited, both competitively and non-competitively, which can reduce their function. And we have cofactors or coenzymes that can facilitate the function of an enzyme by helping to change its shape or providing key molecules for the functioning of that enzyme. So thank you very much for listening. If you have any questions, please bring them to class and I will see you there.